Hello students, today's class is on microscopy. This is a class for nursing students. So what are the learning objectives? At the end of this session, the students will be able to know the definition of microscope, the types of microscope, working of the microscopes, various microscopes and their uses. So first let us look into classification of microorganisms or microbes. The microbes with cellular organization are bacteria, fungi and parasites. Bacteria are also known as they belong to prokaryotes, which have ill-defined nucleus and which do not have membrane-bound organelles, whereas fungi and parasites belong to eukaryotes, which have well-defined nucleus and all membrane-bound organelles like Golgi apparatus, mitochondria, etc. And the microbes without any cellular organization are the viruses. They do not have cellular organization. They depend on either human or plant cells for them to replicate and grow. If you look on to the right side, the scale shows the size of various microbes. The smallest are, if you see what are the smallest objects or things in the universe, they are atoms and molecules. If you look at the microbes, the smallest of the microbes are the viruses. Viruses are the smallest microbes, followed by prokaryotes, that is bacteria, and then a little bigger sized are the eukaryotes, which are fungi and parasites. What is the definition of microscope? Microscope is nothing but an optical instrument that uses a combination of lenses to produce a magnified, that is enlarged image of the objects which are too small to be seen by the unaided eye. Okay, so the resolution power of our eye is just 200 microns. Okay, whereas that of the microscope is 0.2 microns. Okay, so what is this microns? Is a unit of measurement of microbes. It's something 1 micron is equal to 10 to the power of minus 6 meters. Okay, so this word microscope was derived from Latin and Greek words. Micro means small and scopes or scopos is a Greek word which means to look or to see. Looking into types of microscopes, basically there are two types of microscopes. One is light microscope, another is electron microscope. The light microscope uses light source and it uses glass lenses for producing enlarged image of a specimen, whereas electron microscope is using electrons and it's using electromagnetic lenses to produce a enlarged image of an object. Under light microscope, we have bright field microscope, dark field microscope, phase contrast microscope and fluorescent microscope and many other, but knowing these many will be sufficient for now. First, let us look into bright field microscope, which is also known as compound microscope or compound light microscope. It is the most common microscope which is used in microbiology, and it is an optical instrument that uses visible light and glass lenses to produce a enlarged or magnified image of a specimen. And what is the magnification it is producing? It produces a magnification of 1000 times. And how is the image formed? The image of the specimen you're seeing or the microbe you're seeing is darker against the bright background. So this is a uniocular microscope. If you look at this, we have to know the various components or parts of the microscope. The supporting parts of the microscope are the base. You can see the base at the bottom. Look at the bottom, it's the base which is holding the microscope. And then there is an arm, a C-shaped arm with which you can lift up the microscope. And then there is a stage which holds the specimen. And then there is a head along with this black tube which is known as body tube The lower end of this tube is holding one glass lens which is known as objective lens, and the upper end, if you see, of this black tube is holding the ocular lens or the eyepiece, which is another glass lens. These two lenses together produce a magnified image of the object. So apart from this, there is, if you can see above the base, there is a illuminator from which the visible light comes in and enters the specimen. So looking into the components of the microscope, one is a strong metal stand or the base, which you can see at the bottom, above which is a light source or an illuminator from which the visible light comes, and it enters the condenser. The condenser is one which collects the light which is coming from the illuminator and focuses it onto the specimen. Okay, and then above it, above the condenser, is the iris diaphragm, you can see, which regulates the amount of light that has to fall onto the specimen. Now we're saying specimen, where is the specimen placed on? It is placed on a stage which you can see above the iris diaphragm. The specimen is it is smeared onto the glass slide, and the glass slide with the specimen is kept on the stage. Moving on, from the specimen, the light which comes out, which reflects out, it goes into the objective lenses. There are many objective lenses. There is a revolving nose piece which holds many objective lenses of various magnification power. The magnification power can be 4 times, it can be 10 times, it can be 40 times and 100 times. Mainly in microbiology, we use the objective lenses with magnification power of 10x, 40x and 100x. So the light which is reflected from the specimen goes into these objective lens, and the primary image of the specimen is formed. If you're using an objective lens of 10x power, the specimen is magnified 10 times. If you're using an objective lens of 40x, the specimen is magnified or enlarged by 40 times. If you're using an objective lens of 100x, it is magnified by 100 times. Next, where is this going on? Via the body tube. I've shown you the body tube connection, which is going to the eyepiece or the ocular lens, which further magnifies the image. How many times? Only 10 times. The eyepiece has a fixed magnification of 10 times. Okay, and there are various adjustments with which you can adjust the light source, like the iris diaphragm, and coarse and fine adjustments which you can see, through which you can move the objective lens up and down. So this was about the components of the microscope. Now let us again look into what is this eyepiece and objective lens. If there is single eyepiece for the microscope, it is known as uniocular microscope. So you have to use only one eye to look in for the see the enlarged image of the specimen. And this eyepiece is a lens where the viewer looks through to see the magnified image of the specimen, and its magnification power is only 10x. Objective lenses, I told you, are 4 types. It can, depending on its magnification power, but the most common ones which we use in microbiology is low power or 10x objective lens. You can see the image below, which is a yellow band, and then high power, which is having a blue band which has a magnification power of 40x, and an oil immersion lens, which is most important and most commonly used to look into and study the microbes, that is mainly the bacteria, is the oil immersion lens, which has a magnification power of 100x. Okay, so the ones on the right side are the objective lens, which are silver colored. And mainly, what are the objective lenses we use in microbiology? They are low power or 10x objective, high power or 40x objective, and oil immersion objective, which is 100x. The black ones onto the left side are the eyepieces. If it's single, it is uniocular. If two are there, you can see with the both eyes, so it's a binocular microscope. And the silver ones on the right side are the objective lenses. I hope it's clear. It's very important to know what is low power, high power and oil immersion lenses. So now how much magnification is produced? What is the total magnification produced? The total magnification produced is a product of magnification that is produced by the objective lens into the magnification that is produced by the eyepiece or the ocular lens. So if you look at the table below, the magnification produced by ocular lens is always only 10 times. Okay, so where is the what is the magnification of objective? If you're using the objective with 4x power, it only magnifies 4 times. So the total magnification is 40x. Say using an objective lens with 10x, the total magnification is 100x. So it's a product of 10x of objective and 10x of ocular lens, which is producing total magnification of 100x. So you're using an objective lens of 100x, the last one below. So the total magnification is 100x into 10x of the ocular lens, which is a 1000 times magnification, which is produced by a bright field microscope. So in microscopy, there are few important things to be known. One is magnification, that is a product of magnification of eyepiece and magnification of objective lens. Similarly, you have to know what is resolving power. Resolving power is ability to distinguish two objects which are placed beside each other as distinct and separate. Okay, so the resolving power of human eye is 200 micrometers. Okay, whereas that of the microscope is just 0.2 microns. Two objects which are placed at a distance of even 0.2 microns can be seen as distinct and separate, whereas for the human eye, the distance between two objects needs to be 200 microns to make out that they are separate objects. Similarly, other term we have to know is the contrast. Okay, so contrast is, say you're taking a picture with the camera. If it's a black and white picture, there is nothing like, the image is black and white. But when you take a color photo, there's a contrast. Okay, so this contrast in microscope, in bright field microscope, is obtained by using certain stains or dyes which color the microbes. Okay, now let us move on. Let us look into the functioning of the microscope or working of the microscope. Look into the flow chart on the left side. From below, if you see the light source, that is from the illuminator, the light comes up. It is collected by the condenser. It is focused onto the specimen which is placed on the on the stage, and then from there the light which is reflected by the or which comes out of the specimen falls onto the objective lens, which magnifies the image, which forms a magnified image of the object, which is known as intermediate image, and this image is further magnified by the eyepiece or the ocular lens, and then this magnified image can be seen by the human eye. This is the functioning of the or working of the microscope. Let us move on. So I told you how do you provide contrast? By staining the specimen, you can provide a contrast. You can see this image on the right side, which is a stained image, but once you stain, you're killing the microbes. So you can see nice colored microbes, the purple colored bacteria, which are round and which are in pairs, against some material, cellular material, which is stained pink color, and the background is usually white. Okay, whereas if you look at the unstained specimen, you have not used any stain or dye, directly you are seeing under the microscope, this is how it looks. So if you are not staining, the microbes are still live and you can see them moving. So this is the product of magnification. If you're magnifying an image 40 times, so this is how it looks, 40x. Next is 100x, 40 times, 400 times. 1000 times is more clear. You can make out them as separate microbes. So let us look into advantages and disadvantages of bright field microscope. Advantages are, it is used to observe stained microbes, and unstained or live microbes like bacteria, fungi, and parasites can be seen, not the viruses. Viruses are too small to be seen under bright field microscope. Then you can observe their morphology, that is how, what is their shape, are they round, are they rod like, are they spiral, how are they arranged, either are they single, are they in pairs, are they in groups. Then you can observe their motility, how they move, do they move or they don't have motility. Then also you can measure the size of these microbes, known as micrometry. But what is a drawback? You can't see the viruses because they're smaller in size. You cannot observe microbes less than 0.2 micrometers. So you can't see viruses, and you can't see even certain bacteria which are very slender, and which it is like spirochetes. The example is spirochetes. Treponema pallidum is an example of spirochete, which you can't see directly under the light microscope. So that was about one type of light microscope, that is bright field microscope. The field or the background is bright and the image formed is darker over here. Next, let us move on to dark field microscope. So dark field, that is the field surrounding the microbes, is darker and the microbes as such appear bright over here. Looking into the principle, here you use a special condenser. It's the same built like the bright field microscope, but the condenser here is a special condenser known as dark field condenser, which has a stopper which will not allow the entire light coming from the light source to directly fall on the specimen. Only the light rays which fall onto the specimen, they fall on obliquely, and the light which is reflected by the specimen only enters the objective lens, and hence the specimen appears bright against the dark background. So to repeat, here the condenser is a special type of condenser which is having a stopper which will not allow the entire light to fall on the specimen. So only oblique rays of light come and fall onto the specimen, and whichever light which is reflected by the specimen only is entering the objective lens, not the entire light. So the specimen appears bright against a dark background. That's why it's known as dark field microscope. Now let us compare the bright field and the dark field microscope. On the left side you have the bright field. You can see all the light sources, light rays are entering through the condenser and they are falling on the specimen, and then these rays are falling into the objective lens. Look onto your right. Here is a dark field microscope with a condenser. You can see this the purple colored light purple colored dark field stopper, which is not allowing all the light rays to come in. Only some light rays are coming and they are obliquely entering onto the being focused onto the specimen, and only you can see the blue colored rays which are reflected by the specimen are entering into the objective lens to form a brighter image of the microbe against a dark field or a dark background. Okay. So what is the use of dark field microscope? You can observe live microbes. So in dark field microscopy, you're not staining the specimen. You're seeing an unstained specimen. That is the live microscope, live microbes. Okay. So the purpose is, it is used to observe live unstained microbes that are either invisible under light microscope or which cannot be stained by the standard methods like gram stain and all, or which can be distorted by staining. So used to detect thin bacteria like spirochetes, example Treponema pallidum. You can detect the microfilaria, the larval form of one of the parasite known as Wuchereria. You can study the motility of the bacteria and the protozoa. So it's very important. You have to know the principle and the uses of the dark field microscope. It's a very important short note. Now let us move on to So let us move on to phase contrast microscope. Phase contrast microscope, here it is also using optics, which includes a special objective and a special condenser, which is changing the differences in the refractive index between the specimen and the surrounding media into variations in light intensity, thereby producing differences in contrast. So what does this mean? What is this refractive index? So in the specimen, we know that there are certain areas which are denser and certain areas which are thinner. So the areas which are denser, the light rays passing through them will bend a little more than those who compared when you compare with the part of the specimen which is thinner, the light rays pass straight through it, whereas the areas which are denser or thicker, the light rays tend to diffract or bend. So by bending there is variation in the light intensity. So these differences in the thickness of the specimen, or the refractive index of the specimen, or the light bending capacity of the specimen based on their density, is converted into variations in light intensity, and this variation is light intensity is used to produce the contrast. Here also you're not staining with any dyes to produce contrast, but using special optics like special objective lens and special condenser, which will change the differences in in the thickness or the density, which because of which the light diffraction changes, whether they are getting diffracted or they're just passing straight through the specimen varies, which is reflected as variation in the light intensity, which produces the contrast in the specimen. So what are the uses of phase contrast microscope? They used to observe the live specimens. You have to remember, dark field and phase contrast microscope, you're observing only live specimens, you're not staining. So here you can examine the live bacteria, fungi, parasites. You can detect the motility of the bacteria. You can detect even the bacterial components. Within the bacteria, sometimes there's some spores, endospores. There's certain inclusion bodies which can be seen through phase contrast microscope. And as such, you can't see viruses, but to grow viruses, you grow them in, I told you, either animal cells or plant cells. So when you're growing them in animal cells, the viruses tend to produce certain changes in these cells. The changes in the cells, known as cytopathic effect, can be seen through the phase contrast microscope. So this is the image of the phase contrast microscope, how the background and the specimen looks like. So here you can see the difference between the bright field microscope, okay, and the phase contrast microscope. Let us move on to fluorescent microscope. Here the light source is not the visible light like the above microscopes, that is bright field, dark field and phase contrast. Here the light source is the UV rays, and here the dyes and the stains which we are using are the fluorescent dyes, like you use a fluorescent markers. So these dyes are fluorescent dyes which produce fluorescence. Examples of these dyes are ecridine orange, auramine and chalcofluor white. So what you do is you use these dyes to stain the specimen, and when the UV rays fall on this fluorescent stained specimen, they absorb the UV light, which is short wavelength light, and in turn they emit a longer wavelength light, which is visible light, which can be seen with the by the human eye, and the specimen appears luminous, that is fluorescent, against a dark background. So this is the working of fluorescent microscope. You really don't need to know, but for those who are interested how it functions, you can know it. The light source is the lamp which is giving the UV light. Then you see the blue light, which is a UV light. Take this blue light as UV light. It is falling onto the specimen. So specimen is stained with the fluorescent dye. So it absorbs these short wavelength UV light and emits a longer wavelength visible light, which is this red light, red arrow, which is going up, and you can see the fluorescent or a luminous image of the object can be seen. Okay. Now, what are the uses of fluorescent microscope? Useful for diagnosing variety of infections, bacteria, fungi, and protozoa. In bacteria, it's very useful for identifying Mycobacterium tuberculosis, for identifying fungi like Cryptococcus, Candida, for identifying parasites like Trypanosoma, Nemocystis, and Cryptosporidium, and many others. There is one technique here, a different technique than discussed above, known as immunofluorescence. You will understand it in subsequent classes, where you can detect even viruses by this method, like herpes virus and influenza virus, and you can also detect bacteria which are difficult to observe under other types of microscope, like syphilis bacteria, the spirochete I told you, the Treponema pallidum, and the bacteria chlamydia, which are usually difficult to grow as well as to see under the routine microscope, can be seen by fluorescent microscope. So these are the images. If you use a fluorescent dye acridine orange, you can see that these trypanosoma parasite is appearing oranged against a dark background. Similarly, if you're using rhodamine, the bacteria appear, the microbes appear red against the dark background. And when you're using chalcofluor white, here it's a cryptococcus which is appearing blue against a dark background. So those who are interested to know what is immunofluorescence, in immunofluorescence what you do is, usually what you're doing for fluorescent microscopy, to the specimen on the slide, you're using fluorescent dye to stain. But here, instead of using fluorescent dye directly, the specimen is what? It's an antigen. It has variety of antigens. So externally, what you prepare is an antibody against this antigen, which is tagged with a fluorescent dye. Okay, so when you add this antibody which is tagged with the fluorescent dye, it goes and binds to these antigens on the microbe, and then under the fluorescent microscope you can see that the microbe material is fluorescing. It's luminous against a dark background. Okay, particularly useful for seeing herpes virus inclusion bodies. Now let us move on to a different set of microscope, electron microscope. Here you are not using any visible light, but you're using beam of electrons, and here the lenses are not glass lenses, they are magnetic, electromagnetic lenses. So the beam of electrons, they pass through the specimen, then through a set of magnetic lenses, that is the objective and the projector lens, okay, producing a magnified image of the object, which is seen only on the fluorescent screen. You do not see through the naked eye. And what is the amount of magnification electron microscope produces? It is 100,000 times. The light microscope was only 1,000 times, but the electron microscope magnification is 100,000 times. So you can also see viruses under electron microscope. You can see bacteria, fungi, parasites. Apart from this, even the smallest viruses can be seen under electron microscope. Basically, electron microscope is again of two types: transmission electron microscope and scanning electron microscope. If you see the working, so just look at this flow chart. So there's a tungsten filament which generates electrons. This entire system will work in a separate room. Okay, it needs a separate room. It's a costly affair, because there should be total vacuum, because using electrons. So electrons are generated, they pass through the condenser, they fall onto the specimen. From specimen, they further pass through the objective lens and the projector, which produce a magnified image which can be viewed on the fluorescent screen, not with the naked eye. So this is the working of electron microscope. So what are the advantages? It is useful for visualization of microbes which are even less than 0.2 microns, like viruses. So it allows visualization of internal structures of the microbes. Also scans the surface of the objects to give three-dimensional view of their structure. So you can see one example, Ebola virus. This is a three-dimensional view of how Ebola virus appears. Now let us look into the differences between the light and the electron microscope. Light microscope is what is a light source? Is the visible light, whereas in electron microscope you're using electrons. And what is the resolving power of light microscope? It's only 0.2 microns. Whereas electron microscope, it can see even the microbes which are less than 0.2 microns It is even up to 0.5 nanometers. Nanometers is a measurement which is smaller than microns. Okay, and the magnification produced by light microscope is only 1000 times, at the max 1500 times, whereas electron microscope is 100,000 times. What are the lenses used in light microscope? Glass lenses, whereas in the electron microscope they are electromagnetic lenses. And in the light microscope you can either focus the image on the screen or see with the naked human eye. Okay, whereas in electron microscope you can't see with the naked eye. It has to be, the image has to fall on the fluorescent screen and you can view it. Light microscope is cheaper to use, easy to operate. It does not need a separate room. Can be moved from one place to other. It is portable. Whereas electron is very expensive. It is needs a particular technical skill and needs a special room, because we're using electrons. It should be vacuum packed. It should run in a vacuum. In light microscope, you can see specimens which are living and dead, whereas usually in electron microscope, the specimens are dead because it's vacuum. Okay, so I hope the differences are clear. Thank you.